Welcome to Dr. Balga's podcast for the Curious Doc. Today is 21st December 2025. This videocast is on a science article published 3 days ago, 18 December 2025, titled A Cellular Basis for Heightened Gut Sensitivity in Females. There's an accompanying perspective on pathways of pain in the same issue of Science, with the subtitle Estrogen tunes a crosstalk between hormone secreting cells in the mouse gut to regulate visceral pain. Visceral pain disorders such as irritable bowel syndrome disproportionately affect women, suggesting gender specific gut brain mechanisms. The hypothesis of these investigators was that estrogen enhances gut pain via epithelial neuronal signaling in the colon. They used mouse models, including intact females, overectomized females, and males. Methods: ex vivo mucosal afferent recordings, visceromotor response testing, intestinal organoids, and RNA sequencing. Sample size: typically 5 to 22 mice or afferents per experiment. Cellular assays: 10 to 25 cells. They used statistically two-way repeated measures of analysis of variance, t-tests, and mixed effects models. What did they find? They found that estrogen increased visceral sensitivity. Removal of ovaries markedly reduced the responses. Mechanism: estrogen receptor alpha signaling in L cells increased peptide YY release. Downstream, peptide YY activated enteroendocrine cells, increasing serotonin and pain signaling. The key statistics for estrogen effects: p-value less than 0.0001. Peptide YY restoration: p-value between 0.0005 and 0.001. Limitations of the study: primarily mice data. Human validation is indirect. The translational gap, the study tells us, is hormonal cycling and diet interactions need prospective clinical studies. The accompanying perspective in the same issue of Science 3 days ago talked about the context. Women experience greater visceral pain. Hormonal modulation is a key biological contributor. Their perspective, of the authors: estrogen orchestrates a paracrine dialogue between peptide YY secreting enteroendocrine cells and serotonin-releasing enterochromaffin cells in the colon. The mechanism: estrogen receptor alpha acts on peptide YY cells, indirectly amplifying serotonin signaling to mucosal sensory neurons. Integration: nutritional cues, gut microbiota-derived metabolites, and sex hormones converge on this epithelial pain circuit. Implications: explains menstrual cycle variability of abdominal pain and supports sex-specific, precision approaches to visceral pain management. The outlook: human validation is immune contributions need to be determined, and microbiome hormone interactions remain key unanswered questions. Clinical pearls: women with functional abdominal pain may have hormonally amplified gut sensitivity. Estrogen influences pain locally in the colon, not just centrally. Dietary fermentable carbohydrates may worsen symptoms by fueling this pathway. Serotonin antagonists may blunt estrogen-driven visceral pain responses. These are some key concepts related to this science paper. Hit pause and listen and read at your own leisure. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.